let's move on to the next session, the third plenary. On protecting transplant patients, how do we optimize immune response to vaccination? May I invite the speaker, Dr. Dorry Segev. I invite the chairpersons for this plenary session, Dr. Dhananjay Agarwal, Dr. Tushar Dige, and Dr. Georgie K. Nainan. Over to the chairpersons.
Welcome to all audience and my colleagues, chairperson. Now, next talk is on the protecting the transplant patient and how we can optimize the patient's protection, particularly in terms of the vaccination. And this is more relevant in terms of the COVID vaccination. Now, I invite Dr. Dorry to deliver his lecture on this excellent topic.
Please start recordings.
Thank you for the opportunity to participate in this exciting conference. It's nice to see so many esteemed colleagues and fantastic lectures on the schedule, and it's truly an honor to participate. I wish we could do this in person, but hopefully in the coming months we'll be able to have in-person congregations. I was asked to talk about protecting transplant patients. How do we optimize immune response to vaccination? And perhaps I will subtitle this Lessons from a Study of 8,000 Patients We Never Met in Person, and I'll talk about why that's important later. So the COVID pandemic hit very hard on the transplant side of the medical world. Our recipients have a higher risk of disease acquisition and a higher prevalence of comorbidities, leading to a higher risk of morbidity and mortality, which we all saw in our clinical practice. Now, the vaccines were supposed to be the miracle that solves the pandemic problems, but unfortunately, patients who were taking immunosuppression were excluded from the clinical trials. The vaccine responses require an immune response, and as a result, because our patients are immunosuppressed, in general, there are lower vaccine responses in transplant patients across the board. And so when the new vaccine came out for COVID, and in particular those using the new platform of mRNA vaccine administration, we wanted to understand the safety of vaccines and the immunogenicity in transplant patients. We started our study in December 2020 to understand these issues, but this was unfortunately the height of the third wave in the United States, which, as many are aware, was an incredibly dangerous time for anybody to leave their house, let alone for transplant patients to leave their house. So we had this dilemma of how to do a study on many patients and to keep them safe. The rollout of vaccines was prioritized in the United States, which means that very few patients from each transplant center initially had access to the vaccines. That was prioritized to healthcare workers and frontline people, and so we had to recruit nationally using social media. We generated a very high volume of recruitment, more than 1,000 patients enrolled in just the first week alone, and each of these patients had multiple reactogenicity visits and immunogenicity blood draws, and we had to do all of this in the safest way possible in what I call research in 2021. So a new sort of approach to doing research. As I present the data from our studies, I will also show you the photos of the people who contributed to these, and one of the people contributing dramatically to the design of our study was Jackie Gronzik-Wang, one of our kidney transplant surgeons. We used, to keep people from having to leave their house for blood draws, we used a painless capillary blood collection device that you see pictured here that was able to draw about 200 microliters at a time from our patients. And what we would do is we would mail them their devices, and then when it was time for their next blood draws, they would use this device, put the tube in the mail, and send it to us. So none of our study patients ever had to even leave the house and put themselves at risk during this very dangerous time. Very early on, by end of January, we already had data published in the literature at the beginning of February on safety of vaccination, which we updated by April to include 742 patients. This is work that Michael O, one of our trainees in the lab, did, showing a relatively reasonable safety profile of vaccination in transplant patients to allow us to spread the word to our transplant patients that it was safe to get vaccinated and to give them a sense of what expectations they should have from vaccination. Of course, more importantly were our early findings that the immunogenicity of the vaccine was suboptimal. This is work done by Brian Boyarsky, who's shown here. Originally in March in JAMA, we published data on 436 patients who had antibody detectable in less than 20% of those who received the first dose of an mRNA vaccine. And even in ones who had detectable antibody, the typical antibody levels were quite low. This was in stark contrast to 100% of immunocompetent participants in the randomized trials that sort of established the efficacy of these vaccines, with both of the mRNA platforms. So immunocompetent people, 100% detectable antibody after dose 1. Transplant people, fewer than 20%. This is when we knew by March that there was going to be a problem with vaccination of transplant patients. We found that those on an anti-metabolite were 1/3 as likely to have antibodies as those not on an anti-metabolite. And to put this into perspective, that means that only 10% of people on anti-metabolites had any detectable antibody to the first dose of the vaccine. Older people were less likely to have detectable antibody. And then those more than 6 years out for transplant or those who received the Moderna platform were more likely to develop antibodies after the first dose. Obviously, in March, when we sent our dose 1 data to JAMA, they said, this is important to publish, but please do send us dose 2 data. And by May, we had published dose 2 data on almost 700 patients who received both doses of the mRNA vaccine. And things got better. We were able to detect antibody in 57% of the participants, which is better than less than 20% of the participants, but still 43% had no response. And even those who had a response in general had a much lower response than we were seeing in the general population of people who are immunocompetent. You can see here the blue lines are people who had some response to dose 1, and they all boost very nicely after dose 2, but the overwhelming majority had no response to dose 1, and in response to dose 2, half of them still had no response. Some of them were at the top of the charts and had good response, but most of them had a antibody level that was somewhere in the middle and much lower than that seen in immunocompetent people. So still a major problem for about 75% of transplant recipients after complete vaccination. In this cohort of people who got 2 doses, again, anti-metabolites were associated with a lower probability of developing antibodies. And those who were further out from transplant and who got the Moderna were associated with a higher likelihood of developing antibodies. We looked at this specifically in thoracic transplantation, where immunosuppression is quite different, and also the implications of the disease are quite different. This is work by Andrew Hallett from our group. Among heart patients, 44% were non-responders compared to lung patients where 58% were non-responders. But in both groups, 10% or fewer managed to reach that high boosted level of antibody response. So again, even more worrisome in thoracic transplantation. Less worrisome in liver transplantation, again, because in general they take less immunosuppression. Only 20% were complete non-responders, although still have had a weak response to the antibody. This is work by Ali Strauss, one of our hepatology fellows. When we took all of the results from more than 1,000 patients who got 2 doses, and we put them into a machine learning exploration process, work by Sanjay Bay in our group, we were able to identify which variables were most important in their relationship to an antibody response, and those were MMF, time since the transplant, and age. You see, there are other things like liver transplant, the platform that they got, lung transplant, etc., but those all comprised each less than 5% of the importance in the explanatory power of the model. The most important predictors were those top three, MMF, time since transplantation, and age. Obviously, our study was not the only study to report this. After we reported our findings, many other groups from across the world reported suboptimal immunogenicity from mRNA vaccines or other vaccine types across the entire spectrum of transplant patients. Initially, there was a concern that what we were seeing were just antibody levels, and did they really have any clinical meaning? Andrew Caraba in our immunology lab has started to look at the association between antibody levels as measured and reported in our papers and neutralization assays. And here you see a nice correlation, especially beyond an inflection point of what we would consider to be high positive, between measurable antibody levels and neutralization against both wild type and as well as variants of concern. And then in the general population, which we are now in the process of looking at in transplant patients, but in the general population from one of the very large randomized trials, it was found that neutralization levels against spike and other components of the virus were inversely correlated with risk of a COVID-19 breakthrough infection, with these hazard ratios per 10-fold increase in the marker level, meaning that antibody levels were associated with neutralization, which were then also associated with clinical protection. So there does seem to be some evidence of a clinical correlation between what we're measuring as antibody levels as well as clinical protection. This manifested very clearly when we studied across 17 hospitals 18,000 fully vaccinated transplant recipients, and we looked at reported breakthrough infections as well as breakthroughs with associated hospitalization and death. And if you compare fully vaccinated patients to fully vaccinated general population, as reported by the CDC during the same time periods, there were 82 times higher risk of a breakthrough infection and 485 times higher risk of a breakthrough infection with associated hospitalization or death. So that's really important because it points out, it gives us a clinical dimension for what we were finding with antibody levels. And it also points out that the risk of death from a breakthrough infection in a transplant patient is about 9%, which is very, very high and very different from the conventional wisdom that has been passed that, sure, you might get a breakthrough infection if you're fully vaccinated, but it'll be very mild. That is not the case in transplant patients. This was breakthrough infection work shared with me kindly by Vivek Kute, who published in Transplantation with some colleagues breakthrough infections in transplant recipients across the Oxford AstraZeneca platform as well. So this is not specific just to mRNA vaccines, but also to other platforms of COVID vaccination. And as you can tell from this visual abstract, these patients did not do particularly well when they got a breakthrough infection. Those on Bela seem to do much worse than those on other immunosuppressions. In fact, after dose 1, we see antibody in 0%, after dose 2 in only 5%, and then even after dose 3, we see very, very little detectable antibody in patients taking Bela. So I think this population, plus some other subgroups, will be refractory to active immunity activated by vaccination. In our country, those who got the Johnson & Johnson platform did worse than those who got the mRNA platform. You can see in orange those who got the J&J. Almost all of them were negative, and even those who were positive had very, very, very low levels of antibody compared to much fewer who were negative and a much broader range of antibody detection in those who received the mRNA vaccine. So while mRNA platform is new and wasn't particularly well known before 2021, it seems that it is a better platform, a more successful platform in transplant patients. Pediatric patients also do better than their adult counterparts. Carolyn Chin, one of the trainees in our group, studied 52 COVID-naive pediatric patients ages 12 to 18, and after dose 1, 57% were positive, and after dose 2, 73% were positive, with quite high titers after dose 2, much more comparable to maybe those who are immunocompromised for autoimmune diseases and things like that, much better than what we saw in adult transplant patients. The reasons for this are still being explored, and I'm sure will be very, very interesting. The other group that does better are those who are convalescent, which means those who had COVID-19 in the past and then got vaccinated. If you were a transplant patient who had COVID-19 and then you got your first dose of vaccine, 90% of them had detectable antibody compared to only 18% among those who were COVID naive. And what that shows us is that transplant patients can ultimately create an immune response to COVID, although the immune response that is activated by convalescent disease, by natural disease, seems to be quite strong. One of the reasons for this could be the fact that we reduce immunosuppression when somebody has a COVID-19 infection in a transplant patient, and perhaps that period of reduced immunosuppression helps with immune response. So what should we do knowing that so few of them have a good antibody response after 2 doses? Brian Boyarsky looked at the kinetics of these antibody responses to ask the question, if it's low after 1 month, maybe 3 months after the second dose, it would become much higher. And it turns out that that's not really the case at all. Most who start negative at 1 month stay negative by 3 months. Most who start high positive at 1 month stay high positive by 3 months. So just waiting does not seem to be a viable solution. One other solution would be to continue to give more doses. In April and May in the United States, when there was frustration about the suboptimal immunity generated by 2 doses, there were a number of patients who went and got third doses on their own and reported that to us, and we were able to test their pre- and post-dose 3 immunity. And Bill Werbel here in our group was able to show that even among those who started negative, 1/3 demonstrated a conversion to positive after a third vaccine dose. And those who started low positive, all of them boosted to high positive after a third dose with tolerable reactogenicity in a relatively safe manner, showing that third doses might be the answer for some transplant patients. As we've studied this more in more patients, we have also again seen almost 50%, more than 50% response among those who start negative, and almost 100% among those who start low positive will reach a good level of immunity after a third dose. There have been other reports from France and a randomized trial from Canada confirming these findings as well, and they have impacted national and international policies. You can see here, again, this is unpublished, please don't share this, but these are data from our dose 3 study, where you can see the antibody levels prior to dose 3 and then the rise in the antibody levels after dose 3. And you see that anyone who starts under 50 in what we call the low positive range, all of them make it to a high positive range after the third dose. And many of those who start negative also reach quite high levels of antibody after a third dose. These data, the U.S. data in combination with the confirmatory evidence from other countries, led the FDA and the CDC in August to authorize third doses for immunocompromised people, including those who were transplant recipients. And obviously that has been a huge game changer for the landscape of immunization among transplant patients in the United States. But you notice that even among those who got third doses, many of them still have relatively low antibody titers after that third dose, which we would guess then would correlate with relatively low neutralization assays as well as clinical protection. So what do we do for them? Well, there are two sayings that we could consider, two adages. One is, if at first you don't succeed, try again. Maybe we just need to continue to prime their immune system, and ultimately they will cross over to a point of detectable antibody. The other adage is attributed to Einstein, which is insanity is doing the same thing over and over again and expecting different results. And it is quite possible that there is some reason why these patients don't have an immune response, and that continuing to give them vaccines will not help at all. We recently published this. This is work of Jennifer Alejo from our group. We did look at those who got fourth doses, and it turns out that maybe both of those sayings are correct. About half of them did manage to boost, and about half of them did not. And so I think we can conclude that there are going to be some patients refractory to vaccine immunity, but for some, we just need to continue to prime the immune system until we see a good response, knowing that the early primes are generating some sort of an immune response in those patients. This work drew the attention of the NIH, and in particular of Dr. Anthony Fauci, who runs the branch of the NIH interested in immunosuppression and the immunocompromised, and we were funded to launch a large multi-center national trial where we are now going to investigate booster doses with or without immunosuppression reduction, as well as the role of homologous versus heterologous boosting. And in addition, in all of these studies, we're going to better understand the deeper immunology, including neutralization, but also T-cell repertoire, T-cell activation, memory B cells, etc. And I'm hoping we already finished the pilot for this study. We finished recruiting for that a couple of weeks ago, and we are about to start recruiting for the first randomized component of this trial. And I'm hoping that in the next few months we'll be able to have a lot more information and insights to share from this effort. So from a 10,000-foot view, we might say that vaccines have a good safety profile in transplant recipients, particularly the mRNA platform that we've been able to study. Poor anti-spike antibody responses after vaccination in our patients, better in liver and in pediatrics and in those who are convalescent, worse in those taking antimetabolites and Bella, and worse in those who received the J&J vaccine. We do have evidence from transplant patients that antibody correlates with neutralization, and then we have evidence from the general population that neutralization also correlates with clinical protection. We also have a clinical dimension in our own studies with substantially higher rates of breakthrough infections, and in particular, breakthroughs associated with hospitalization or death. We have found some improvement with booster doses, but there are some patients who will no doubt need other immunity strategies, for example, passive immunity with monoclonal antibodies and things like that. And those are now being tested in immunocompromised populations, and hopefully we'll have a good answer for that soon. From a 50,000-foot view, we might conclude that we can run a study of thousands of people who we've never met in person, and this has major game-changing implications for how we do research and practice medicine in the future. We can interview patients from home. We can even do blood draws from home, and this will impact longitudinal high dimensional surveillance both in detailed granular research studies, but also in clinical practice when monitoring higher risk patients with chronic diseases like transplant patients. In India, Vivek Kute was kind enough to share with me a few slides that COVID vaccination began in January, quite similar to the United States. And as of the end of last month, over 883 million doses were administered, mostly through the Oxford AstraZeneca platform named Covishield, and then through a locally developed platform from Bharat Biotech named Covaxin. There have been new editions of the Sputnik vaccine, Moderna, Johnson & Johnson, and then ZyCoV-D, another locally developed vaccines, and those are currently undergoing clinical trials. And the Indian guidelines for COVID vaccination in transplant recipients have just been published. And again, thank you to Vivek Kute for sharing with me this slide summarizing the vaccination recommendations, ideally prior to the transplant or sometime after the transplant, after induction immunosuppression has waned, and then the two common vaccines available in India and their dosing recommendation. So the booster doses have not been well studied yet in India, but hopefully I'm looking forward to seeing some data come out of India to see if these additional platforms benefit from boosting in the same way that what we have seen in our platforms have benefited from boosting. I'd like to thank my epidemiology research group in organ transplantation. I showed you the pictures and gave you the names of the people driving a lot of this research, but this is a huge team effort. We have a team of about 100 people who've been working to better understand COVID in transplant patients and in particular vaccine responses in our patients and other immunocompromised populations. So a major acknowledgement to all of the people working on this. And again, my thank you for the opportunity to participate in this conference, and my best wishes for a safe and quick emergence from this pandemic and the opportunity to congregate in person again in the future. Thanks.
Thank you. It was an excellent talk, and thank you for sharing the US data to us. Though COVID is a preventable disease to a great extent, this post-transplant patients have got poor antibody response, and we understand that even after 2 doses, your data showed around 57% protection only. See, this is compounded by the fact that the antibody level drops with time. That's a matter of concern. We need more data, especially in our patients, Indian population, regarding this. As our President Jay Prakash said, despite 2 vaccinations, most of the people don't know what their antibody level is. We have to continue with the standard precautions in the prevention of COVID. These are my comments. Thank you. Dr. Tushar?
Sir, has been a very excellent talk, and I thank Dr. Dorry for a very informative talk, and I thank the organizers for organizing such a beautiful conference. With this, Sir said we need more data, and we hope that all our collective efforts will create more and more data and more papers. With this, I want to thank everybody, and may I ask that the session is closed and hand over to the next talk. Thank you.
And we have a few questions from Dr.
Yes,
Dorry.
your comment, Dr. Dhananjay Agarwal, because this is a recording, so question is not possible.
We have listened Dr. Dorry at a different platform in India, and a lot of time he gives a very important information to us. So I have 1 or 2 questions that after 1 dose of vaccination, can we go out for the transplantation in our context? Though we had a discussion and other thing. Second question, after vaccination, transplantation, at what time we should go for vaccination?
Dr. Dhananjay, this was a recorded lecture. So
Okay.
a question cannot be answered. Okay. This was a recorded lecture.
Thank you, sir. Excellent information to the audience. And particularly, I want to extend the thanks from all my residents also that in our department we have conducted the mini session of the conference. We have a conference in our lecture theatre and everybody is attending it. Thank you, sir.